Hi everyone, this is the Edexcel International A Level Biology past paper. This is June 2024 and this is Unit 2 or Paper 2. This is Part 2 of the past paper walkthrough. So if you want to check out Part 1, you can. The link is in the description box below. All right, let's continue on from question 5. All right, question 5. The photograph shows a fruit fly. The gene for eye color in this species of a fruit fly has multiple alleles. Homozygous fruit flies have 2 identical alleles for eye color. The table gives some information about these alleles. Okay, so you can see the different alleles there. All right, these alleles are found in the same locus. State what is meant by the term locus. So this is a 1-mark question, and this is what I wrote. It is the position of a gene on the chromosome. So that would be 1 mark. All right, explain how these alleles can produce fruit flies with different colored eyes. All right, so this is what I wrote. Different alleles would have different DNA sequence. So that would be 1 mark. So for example, let's say a person has A, T, C, G, G, A as their sequence, and another person has A, T, G, 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 A. So it's different at this particular position. So that would mean 2 different alleles, right? And because the DNA sequence is different, then that could code for different amino acid sequence, which results in different proteins. So that's the 2nd mark. Another thing you can also say is talk about incomplete dominance. So here's an example with flowers. So here, you can just sort of ignore the alleles, the R1 and the R2, but basically you've got red flowers and white flowers, and then if the 2 alleles come together, you'd form an intermediate. So between red and white, you've got pink. Okay, and then that would be the 3rd mark that you can add. Okay, let's scroll on to Part B. The development of an embryo into either a female or a male fruit fly is determined by 2 different proteins. The female-specific DSXF protein leads to the development of a female fly, whereas a male-specific protein leads to a male fly. Both proteins are the product of the same gene. This part is important to note down. Explain how this gene can give rise to either female or a male fly. So because we're talking or referring to the same gene, we would kind of assume that the DNA sequence is the same. So in this case, we'd have to talk about alternative splicing, which is something that happens in eukaryotes, and it happens right after transcription takes place. So this is an example of post-transcriptional modification. So you can see the DNA consists of exons, and then this little gap, these sequences here in between are referred to as introns. And so what would happen is you'd form the pre-RNA, which is this strand over here. The introns are removed, and some of the exons are removed in order to produce the mature mRNA. So you can see for protein 1, they've included exons 1, 3, and 5. But for protein 2, they've used exons 2, 3, and 4. And protein 3, they've used exons 1, 2, and 5. So for the same gene, you can make different proteins. So we can say that once the gene is transcribed to form pre-mRNA, it then undergoes alternative splicing. Let me just write that down. Alternative splicing, or you can say post-transcriptional modification. And then we can say the introns are removed. That's another mark as well. And some exons are removed as well to form the mature mRNA. So that would be 3 marks so far. The mature there should be an M here. The mature mRNA is then translated at the ribosome. So that's another mark. And that would result in proteins with different amino acid sequence. So that's the 5th mark. And then you can even elaborate and say the proteins can act as transcription factors which switch on genes related to primary male or female sexual characteristics. So again, primary sexual characteristics refer to characteristics that show up when an organism is born. And that would be the marks for this question. Okay, part C. Histone modification can affect gene expression as shown in the diagram. So you can see when there's methylation, it's more tightly packed. You can see that here. But when there's acetylation, they're more loosely packed. Explain how histone modification can affect gene expression. Use the diagram to support your answer. All right, so first is an explanation on how these modifications can occur. So histone methylation is when a methyl group is added to the amino acids on the histone protein, so that will be 1 mark. And then you can talk about how it causes the DNA to become more tightly packed. So that would be the 2nd mark. Now, because it's more tightly packed, it results in the gene not being expressed. So that would be the 3rd mark. And then you can talk about acetylation, whereas on the other hand, an acetyl group is added to the histone protein. This would actually be the same mark as this one that we mentioned earlier. This causes them to become more loosely packed. Again, this would be the same mark as the one that we mentioned earlier, which would cause the gene to be switched on. So that would be 1 mark. Why? Because the RNA polymerase is able to bind to the DNA. Okay, let's move on to the next question. Water is important for plants. There are pores in the surface of the leaf which are formed by guard cells. Changes to the guard cell causes the pore to open or close. The diagram shows the guard cells on the same time of day from a wheat plant without water stress and from a wheat plant that has water stress. So without water stress and with water stress. So this is without and this is with. When a plant is not given sufficient water for a period of time, it develops water stress. This can have several effects on the plant. So I'll just explain why this happens. So if the plant is given sufficient water, what happens is that water would go inside of the guard cells, and that would cause the cell to become turgid. And when the cell is turgid, it just so happens to look like a U-shaped or a banana shape. Now, when there's not enough water, water would leave the cells or the guard cells, and that would cause the cells to become flaccid, and so that's what the guard cells look like with a lack of water. One effect is to close the pores of the leaves. This occurs due to changes in the guard cell. Part 1, name the structure labeled X. So X is pointing to the vacuole. So I'm just going to write vacuole. All right. Part 2, suggest why the closing of the pore reduces water stress in the plant. So what happens is when it opens, it actually allows for water vapor to leave the leaf, and that is called transpiration. When it's closed, it prevents the loss of water through transpiration. All right, so that's what I wrote here, to prevent the loss of water via transpiration. Let's move on to part B. Another effect is a reduction in the absorption of mineral ions, for example, magnesium ions. Magnesium ions are needed for the formation of chlorophyll. The effect of water stress on the chlorophyll content of a wheat plant was investigated. So you can see with water stress, there was less chlorophyll. So this part, it says, which is the percentage change of chlorophyll content when the wheat plant has water stress? So the chlorophyll content with water stress, it looks like it's about 3.2 minus this one would be 1.7. We would divide it by the original, so 3.2, and times by 100. And the answer would be 46.9%, and it's a decrease. So the answer is B. All right, part C. The graphs shows 3 more effects of water stress on wheat plant. So the first graph, graph A, is about mean percentage survival of wheat plants. Graph B is the effect of water stress on cellulose. Graph C is the effect on the root length. All right, to deduce how water stress could affect the growth of the wheat plant, use your use information from the whole of question 6 and your own knowledge to support your answer. So in order to answer this question, you need to talk about the trends that you see from the graph. So you need to describe the data, and you need to link that to how you would think that would impact the growth of the wheat plants. And remember to read the entire question. So for example, earlier here, they mentioned that magnesium ions are needed for chlorophyll formation. So that's important to keep in mind. Okay, so this is what I wrote. So first I talked about the data. So if you look back at the data, with water stress, there's lower chlorophyll content, lower mean survival percentage, lower cellulose content, and lower root length. And that's what I wrote. According to the 4 graphs, when there is water stress, you've got lower percentage survival of wheat plants, lower cellulose content, shorter root length, and also lower chlorophyll content. And then I mentioned the statistical data, or the statistical analyses. So this graph does not have any error bars. This graph also do not have error bars, but these two over here, they do. So I would write, however, this comparison is only statistically significant with the mean cellulose content and the mean root data. The other 2 graphs do not have error bars. All right, then I would go on to explain, so using my own knowledge and the whole of question 6 as to why we see what we see. So shorter root length would mean fewer ions, like magnesium ions, would be absorbed. So that would be one point you can say, which would lead to less chlorophyll being formed, because remember earlier they mentioned that magnesium ions are used for chlorophyll formation. Less chlorophyll would mean less light being absorbed for photosynthesis. And less photosynthesis would mean less glucose being produced. Now, how do we link that to cellulose content? Well, beta-glucose is used to make cellulose, and this would cause an overall decrease in cellulose content. So these are some points that we can say. Cellulose makes up the plant cell wall. As there is less cellulose, that would cause the plant cell walls to be weaker, which would mean there is less support as the plant grows, and that's probably why the percentage survival of wheat plants are lower. All right. Let's move on to question 7. The photograph shows a mother and a baby Malayan tapir. They live in the rainforests of Southeast Asia. Each gamete of a Malayan tapir contains 26 chromosomes. Part A. The body cells of the mother Malayan tapir have some genetic similarities and differences from the cells of her baby. Explain why the body cells of the mother and baby have genetic similarities and differences. So first I talked about the similarities. So there are genetic similarities because the baby inherits half of the chromosomes from the mother. So that would be 1 mark. And then I mentioned the reason why there are genetic differences is because the other half of the chromosomes are inherited from the father. And there may have been mutations that took place when producing the female gamete, so the baby might have inherited those mutations. Okay, let's move on to part B. Biodiversity within a species can be measured using genetic diversity. 3 melanistic Malayan tapirs have been discovered to have black fur all over their bodies. Melanistic tapirs have 2 copies of the recessive allele for fur color and are homozygous. Scientists want to determine the heterozygosity index of the Malayan tapir species. What is the equation? So the answer for this would be A, because it's the number of heterozygous individuals divided by the total number of individuals in the population. All right, part 2, explain how scientists could determine if a change in the frequency of the recessive allele was occurring over time. So we need to talk about the Hardy-Weinberg equation for this. Okay, so I've included the equations for the Hardy-Weinberg equation. We've got P plus Q equals 1. So P is the frequency of dominant alleles, and Q is the frequency of recessive alleles. And then you've got P squared plus 2PQ plus Q squared equals 1. So P squared would be frequency of homozygous dominant, and that's why you square this. And then 2PQ would be heterozygous, and then Q squared would be frequency of homozygous recessive individuals. So the first thing we want to do is, in this scenario, we would count the number of melanastic tapers. So for example, let's say there were 6 melanastic tapers, and then we would count the total number of tapers. So let's say there was 100. So that would give us 0.06 as the frequency of homozygous recessive individuals, because all of the homozygous recessive individuals would be melanastic. And then we want to find the frequency of recessive alleles, so we would just square root this to get Q. So in this case, it'll be like 0.245. And then because the scientists want to determine whether or not there's a change in the frequency of recessive alleles, what they could do is repeat this process over time. So let's say every 6 months or every year. And then afterwards, compare and see if there are any changes. So this is what I wrote. First, count the total number of tapirs in all of the population. So that's the first mark. The second mark is use the Hardy-Weinberg equation to calculate the recessive allele frequency. So I wrote this and then square rooting it to get Q. So that will be the second mark. And the third mark would be to repeat it over time and then compare to see if there are any changes. All right, that would be 3 marks. Part C, the map shows the change in distribution of the Malayan taper over the past 20 years. And then part 1, it says, give one similarity between the current and earlier distribution of the Malayan taper. Okay, so for part 1, I wrote the tapers are still found in Thailand and Sumatra. Part 2, one difference is that the distribution has reduced due to human activity, suggest one reason why human activity has caused this damage in distribution, or this change in distribution. Okay, for this part, I wrote one reason could be deforestation, which could cause the tapers to lose their habitat. So that would be the mark there, and that would cause them to decrease in number. Okay, part 3, a study suggested that the genetic diversity of the Malayan tapers has decreased due to populations being separated from each other. Explain how a captive breeding program could maintain the genetic diversity of Malayan tapir populations. So we know that the Malayan tapirs, you can see that here, are now existing in more isolated populations. So it's important to interbreed those tapirs coming from different populations together, and that could help to maintain the genetic diversity. So first, gather individuals from the separated populations and sequence their DNA to determine individuals that are genetically different. So that would be 1 point there. Then breed the tapirs that are genetically different from each other, especially breeding the tapirs between different populations to prevent inbreeding. So this would be another point. And then I also wrote, make sure to record the which individuals are bred together using a stud book. So that would be the third mark. This ensures that rare alleles are still maintained in the gene pool. And that's how we would maintain genetic diversity. Okay. Question 8. The photograph shows a species of fungus growing on a dead tree in a woodland habitat. Wood contains a high proportion of lignin and cellulose. Fungi can break down molecules in wood by releasing extracellular enzymes. Part A, describe the role of the rough endoplasmic reticulum in the formation of extracellular enzymes. Okay, so this is what I wrote. Translation would take place at the rough endoplasmic reticulum. So that's the first mark. And then after it's translated, then the protein folds into its correct 3D shape. So I wrote correct secondary and tertiary structure. So once that is formed, it will then be packaged into vesicles so that it can be sent to the Golgi apparatus. That'll be the third mark. Okay, part B. Different species of fungus can break down different molecules in wood. The effect of two species of fungus on the percentage loss and mass of wooden blocks was investigated. Species A was grown on one set of wood blocks, and species B was grown on another set of wood blocks. The percentage loss in the mass of blocks were recorded every 4 weeks. All right, and we can see that here. And then part 1, comment on the results of this investigation. This is what I wrote. As time passes, the mean percentage loss for both of these species increases. Right, and we can see that over the 4 weeks, you can see that the general trend is going upwards. We know that species A is much better at breaking down the wood, so the percentage loss in mass is much greater compared to species B. And the difference between them is statistically significant because you can see there are no error bars that are overlapping. All right, so I wrote, as time passes, the mean percentage loss in mass for both species increases. However, the rate of loss in mass for species A increases at a greater rate compared to B. That'd be the second mark, and then you would always talk about the error bars. So in this case, the difference is statistically significant. Part 2 suggests one reason for the difference in breakdown of the wood by species A and species B. All right, so I wrote this is probably due to the different enzymes that are present in both of these species, because the different enzymes would break them down at different rates. All right, part C. The study compared the biodiversity of two woodland habitats. The index of diversity for habitat 1 was 4.2. The table shows the data obtained from habitat 2. And then we have the index of diversity equation that's over here. And then it says calculate the diversity index for habitat 2. Use the table and formula to help you. So here at the bottom, we would want to write out the total. So we're going to use a calculator for this. So 34 plus 6 plus 27 plus 4 plus 9 plus 31 plus 120. That gives us 231. I'm going to do the same with the other column. So 1,122 plus 30 plus, ooh, this one's missing. To find this one out, we can do this. So 27 minus 1. Let me just type that into the calculator. And that gives us 702. So now we can count the total. Actually, I'll just leave that there. So 1,122 plus 30 plus 702 plus 12 plus 72 plus 930 plus 14280. And that gives us 17148. So the capital N over here represents the total number of organisms in the community. So in this case, let me just write this down here. So the total number is 231, and then brackets 231 minus 1. Divided by this is what we just calculated over here. So 17148. And the answer is 3.098. Now, the greater the number, the higher the levels of diversity. So I'm just going to write that there, 3.098. Now, if you recall, the index of diversity takes into account two things: species richness, which is how many different species are present, and species evenness, which is how many individuals are there among the different species and how evenly spread or how evenly distributed those individuals are among those species. Okay, and then the next part is state which habitat has a higher biodiversity, and that would be habitat. Hold on, so habitat 1 was 4.2. Yeah, so it would be habitat 1. All right, this is what I wrote. Habitat 2 has a greater biodiversity because the index of diversity is greater. Sorry, habitat 1 has a greater biodiversity. Okay, let's go to part 3. Species D is an endangered plant species. Explain the process that could be used by a seed bank to conserve this species. Okay, so the first thing to do is to collect seeds from different plants of species D. And then to prevent microorganisms from growing, the seeds are sterilized. And then to check whether or not their embryo in the seeds are viable, the seeds are visualized using X-ray, so that would be a third mark. So to prevent germination from occurring, because if you recall, germination requires 3 things: water, warmth, and oxygen. So to prevent germination from occurring, the seeds are kept in a dry and very low temperatures. All right, that is it for question 8, and that is the end of unit 2. If you have any questions about this paper, you can leave them in the comment section below. Check out the other videos on the playlist or on the rest of the YouTube channel. And that's it. Happy studying. Bye!